Our next major topic today is membrane potential. Before I jump into this, I need to apologize because I just realized as I was switching from recording part 1 to part 2 that I forgot to switch over my audio source. So for the first part today, I was using those first 2 sections today are going to involve a really terrible mic. I'm sorry, the audio quality is going to be terrible, but I switched over to the correct audio source for this one. So, oops, sorry. But anyway, now that we've established the structure of neurons, we can talk about membrane potential, or MP, which is going to be critical for how action potentials work. The starting point for this is that the plasma membrane surrounding all of our cells is a selectively permeable membrane, and that should be familiar to everybody. We talked about this way back in 115. Now, the important thing about a selectively permeable membrane in the context of our cells is that it is polarized. This means there is a difference in charge on either side, and specifically the outside is more positive than the inside. So we have more positive ions on the outside of that membrane compared to on the inside of that membrane in the cytoplasm. And this is really important because this represents potential energy. Membrane potential is a form of potential energy, which means it is the capacity to do work. It means your neurons can accomplish something using that potential energy. Neurons are one type of cell that is called an excitable cell. Excitable cells are cells that are able to rapidly change their membrane potential. We are only going to talk about 2 types of excitable cells in GenBio. We're going to talk about neurons right now, and then down the road we're going to talk about muscle cells, which are also capable of very rapidly changing their membrane potential. The way they do it is going to be very different, but the overall principles are going to be the same. So for GenBio, our excitable cells are going to be neurons, and muscle cells, those are the cells that can change that charge differential very rapidly that's associated with their plasma membranes. We measure this membrane potential in terms of voltage. Voltage is just a measure of the difference in charge on 2 sides of a membrane. And we can measure this with a device called a voltmeter. Now, I don't need you to know like the ins and outs of the voltmeter, but basically you can take a little electrode and put it inside the cell, and you have a reference electrode outside the cell. And by comparing the charge density in those 2 environments, you can tell what the difference is across that membrane, and that allows you to determine the voltage. And again, that voltage is just the measure of that membrane potential. How different are those 2 sides of the membrane? All of the cells in our body have what's called a resting potential. This is the membrane potential of a cell at rest, which just means it's not excited. It's not rapidly changing the concentration of ions on either side of its membrane. So it's just kind of hanging out, doing its thing. Whatever that membrane potential is, we call that the resting potential. So typically the resting potential within the human body is going to be somewhere between minus 60 to minus 80 millivolts. We're going to take the average and just say minus 70 millivolts. And all this means is that there is a greater amount of positive charge on the outside compared to the inside. So the inside of the cell is negative compared to the outside, relatively speaking. So we call it negative 70 millivolts. That's about the average resting potential within the human body. There are 2 main things that are driving this resting potential, and we'll talk about all of them in over the next few minutes here. One of them is something that we've talked about a lot, the sodium-potassium pump. And then working alongside that are ion channels that are open and closed at different times that are going to facilitate different ions being in different places, either inside or outside the membrane. And those 2 things together basically drive our membrane potential. So we're going to kind of build the different components that are driving our resting potential of negative 70 millivolts. And then we'll come back to this figure once we've established all the parts and kind of go through all of them all together. The first component that is driving the resting potential that we're going to talk about is the sodium-potassium pump. So we've got the 2 main ions that we're talking about here in the context of an action potential are sodium, which is Na, that's a positive ion, and potassium, which is also positive, and that's K. So the sodium-potassium pump, we've talked about this before. It is a transmembrane transport protein. It uses active transport, so every cycle costs a single ATP. And you find these all through the neuron. They're just constantly going, constantly cycling. And the way they work is each cycle involves moving 3 sodium out and 2 potassium in. So we have a net movement here where sodium is going out and we're generating a sodium gradient on the outside of a cell, meaning we have lots of sodium outside the cell. And then inside the cell, we're generating a potassium gradient. So we have more potassium inside compared to outside. But because it's a 3-to-2 ratio, we're generating more positive charges outside the cell on net compared to inside. Now here is the way this cycle works. The thing I need you to get here is it costs 1 ATP, it's 3 sodium out and 2 potassium in. An easy way to remember which ion goes in which direction, because it can be hard to kind of keep straight which goes which, just remember that sodium is Na and potassium is K, and you could think, nah, get out. K, come in. And that's how I always remember it. That's the only way that actually got me to remember which ion goes in which direction. So just remember that, nah, get out, K, come in. And it's 3 sodium out and 2 potassium in at the cost of 1 ATP, and that's the operation of the sodium-potassium pump. In addition to the pump, there are ion channels that are driving this resting membrane potential. Most importantly, you have potassium channels that are always open. These are called leaky potassium channels because they're always allowing potassium to diffuse. We just saw with the sodium-potassium pump that potassium is constantly being pumped into the cells, but these leaky channels, these leaky ion channels, are just allowing that potassium to constantly diffuse back out. So even though we're generating a potassium gradient, some of that gradient is constantly going to be discharged through these leaky potassium channels. Now in terms of sodium, that is going to be pumped out of the cell, but there are no leaky sodium channels. So once it's outside the cell, it's stuck there. Put all this together and you have, just through the sodium-potassium pump, you have a 3-to-2 net difference in charge, but then of those potassiums, some of those are going to leak back outside through the potassium gradient, which is going to be independent of that sodium gradient. Remember, diffusion is specific to each substance. So you end up with a lot more positive charges outside the cell compared to inside the cell, and that leads to the resting membrane potential, which again, that is a form of potential energy. So here is the overall figure for the resting membrane potential. This is figure 48.6. And you can see here we have a sodium-potassium pump. So these little circles represent sodium. These little orange squares represent potassium. We have some leaky potassium channels that are allowing the potassium to diffuse back in. I don't know why this sodium channel is here because there are no leaky sodium channels that are operating, but for whatever reason it shows it. But the point is that outside you have a higher density of positive charges compared to inside, so you have a negative resting membrane potential, and that resting potential is about minus 70 millivolts. Of course, in order for neurons to work, we're not going to keep that resting membrane potential the same. We have to change the membrane potential, and there are 2 ways we can do that. The first is a hyperpolarization. This means that the membrane becomes more polarized, so it becomes further from zero, which means because we're starting at a negative point, it's going to become more negative. So in this figure, the y-axis shows you the membrane potential in millivolts. We got plus 50, 0, negative 50, negative 100, and the red represents the actual membrane potential. Right here at the left side, it is the resting potential of about negative 70, and then any change from that makes it more negative, further from zero, that is called a hyperpolarization. So in this example, it hyperpolarizes from negative 70 down to about negative 75 or negative 80 millivolts. So what's going to be the opposite of a hyperpolarization? It's not a hypopolarization, that's what you were thinking. It's actually a depolarization. Depolarization because you are decreasing the magnitude of the polarization, meaning the membrane potential is moving closer to zero. And since we're starting from a resting potential of about negative 70, that means we're going to become more positive. We're going to move closer to zero. So here is an example of a depolarization. We have our resting potential, and then it depolarizes up to negative 40 or something, whatever that would be. The final thing we'll talk about in the context of membrane potentials is the threshold potential. And this is the membrane potential required to trigger an action potential. For most neurons, the threshold potential is negative 55 millivolts. And in this same figure we just saw right here, you can see the dotted line indicates the threshold potential of negative 55 millivolts. What that means is that starting from this resting potential of about negative 70 millivolts, you need to depolarize to negative 55 in order to trigger an action potential. In the next section, we're going to talk about exactly what happens when you do that.